we're going to give you a brief introduction of how to extract really nice, well-isolated units using two methods. The first is manually looking at the data, manual curation, and we're going to be using Phi in order to do this. And then Noam is going to talk about different automated approaches using different quality metrics, and then we should hopefully have a little bit of time for questions at the end. So why do we want to do this at all? Well, spike sorting is never perfect, and you always want to look at your data because you will always have some units that are just purely noise, and of units that are actually have spikes that come from real neurons. These spikes can have false positives where some of the spikes come from one neuron and other spikes come from another neurons. And in this case, you'll want to classify them that way, be able to identify them and try and correct this. And units can also have spikes that are missing, which you also need to try and correct. So spikes that are not detected or that have wrongly been split into different units. So the first thing we're going to look at is noise, which is really pretty easy to spot, which is nice. So real neurons have several properties, which you probably know. The first of which neurons have a particular waveform, which looks something like this. There's usually one really nicely defined trough that's quite large and one smaller peak. And this is true for neurons that are recorded near the soma, for spikes that are recorded near the soma of a neuron. And if you are recorded from other areas like axons, non-somatic areas of a neuron, spikes might look a little bit different with notably a larger first peak. And so whether you want to keep this or not depends on really the questions you want to ask. And finally, because sites in neuropixel probes are so close to each other, you are going to record each neuron on several sites simultaneously. And what's going to happen is that the spikes of that neuron are going to be larger on sites that are closer to that particular neuron. And then as you get further away from the neuron, the spikes are going to get smaller with a smaller amplitude. So you can have this property, which we usually refer to as spatial decay, that should look like this. And noise will not necessarily have these properties. So this is something that's fairly easy to spot. The next thing you're going to want to watch out for is if your unit actually contains spikes that come from several different neurons as opposed to just coming from one specific neuron. And there are also a few things you can look at here. These views also are exactly what you would see if you look at them in a software called Phi, which is usually what you're going to use to look at your data. And we'll do a brief overview of Phi after this. So one of the first things that can clue you in to the fact that there's potentially some contamination in your unit and not all the spikes come from the same neuron is if there are refractory period violations. So usually neurons have a refractory period of about 2 milliseconds, but it really depends on the specific neuronal subtype you're looking at. And so then you can see this in autocurlogram, which is what's plotted here for one unit on the left. So if you look in the autocurlogram at the probability a unit emits a spike as a function of the time elapsed since previous spikes, it should be close to 0. So here is spike count on the y-axis and time is on the x-axis with the center being 0. It should be close to 0 spike counts during those 2 milliseconds, which are denoted by these 2 red bars. And you have these lines usually in Phi. And it makes sense also to look at this relatively to how the neuron is spiking, so the neuron's mean firing rate also. So this is an example of a unit that has really no refractory period violations. And on the right, you can see a unit that has a lot of refractory period violations. And a lot of these spikes are coming from at least more than one neuron because of that. But this is not always the case. So you can also have scenarios where spikes from two different neurons have been wrongly assigned to one unit, and you do not see any refractory period violations. So here what we've plotted is the amplitude scaling factor. This is sort of a proxy for each spike's amplitude as a function of time, with each dot here being a spike. In blue are spikes from the current unit, and spikes from nearby units are plotted in gray in the background. And you can see that at the start of this recording, the spikes seem very separate and distinct from neurons later on in the recording. And most likely these are coming from two different neurons, and these are really two different sets of spikes. But because they don't overlap in time, we're not going to get any refractory period violations. Finally, units can contain false negatives. They can be missing certain spikes for a neuron. So this happens because potential spikes are detected usually in most spike sorting algorithms by a simple thresholding, which is denoted by this line. So anything that goes below or above that line is classified as a potential spike. And so if you have units that are further away from the probe, or even just smaller units, that some of the spikes might not be detected. As you see, like this spike here is much smaller than, for instance, some of these spikes over here. And this can result in a large fraction of a neuron spike just not being detected because they are so small. So here, this is raw data on the left, and on the right is the same raw data, but after running a spike sorting algorithm, and each color is a particular unit's spike. So this, for instance, green shows really nice, well-isolated, high amplitude spike. And this unit, you can see it's all its spike plotted as a function of time and amplitude. And you can see that they all seem to be detected. And if you plot the histogram of this distribution, you can see there's a nice bell curve. In contrast, for another unit, you might have something that looks more like this, where it really seems like there's a cutoff here. And some of the higher amplitude spikes of that unit are being detected, but a large fraction of the lower amplitude spikes are completely being missed. So for this unit, for instance, we think that there's a spike here that's just not being detected because it's so small. And so this particular plotting the amplitude as a function of time and the histogram of the distribution of amplitudes is a really useful tool to try and examine if any of the spikes are not being detected. Finally, there's three other things you really need to look at to try and understand if units have some spikes that are not wrongly classified in another unit. So the first thing could be that there's a very similar other unit. Here what we're plotting is, as previously, an autoparallelogram for one unit. This is an autoparallelogram for another unit. And in white here is the cross parallelogram of these two units. And you can see that, first of all, the two autoparallelograms look pretty similar. And also the cross parallelograms don't have any refractory period violations. Finally, if you plot waveforms, so here we're plotting template waveforms, but you should also look at raw waveforms. You can see the waveforms are also pretty similar. And in this case, these spikes can overlap in time. So these are probably 2 units that should be merged into 1 unit because they are so similar. Something else that can happen is drift, which happens a lot. So again, here what we can see is very similar autocurlograms, and the waveforms are also similar. And the spikes are present at different times in the recording, with 1 unit's spikes being really present in the beginning and the other units more at the end. And in this case, it's likely that because of drift, the neuron moved relative to the probe and then was classified into 2 different units that had slightly different waveforms, but actually these might come from 1 same neuron. And finally, the last thing you would really want to look for here are cells that are bursty. So bursty cells fire in bursts. And here is plotted this neuron's waveform. And you can see the amplitude as you progress along the burst, and the amplitude changes. So if you have a burst of, say, 5 spikes that are really close to each other, the amplitude of that unit will change, which means that very often these are wrongly classified by spike sorting algorithms into different units that you need to then merge back together into 1 unit. Some of the properties that you would typically see in this case are cross-kilograms where 1 unit seems to come just after the other. So 1 unit will spike right after the first unit's spikes, which is consistent with a burst. The waveforms would be quite similar, but one of the unit's waveforms which have a smaller amplitude. And finally, the spikes would be present at the same time, but again, one of the unit spikes would have a much smaller amplitude and might have this waterfall look to it, where each spike in the burst has a lower amplitude. So how would you go about, now that we've seen sort of the really basic beginner building blocks of what to look for in your data to try and determine what is noise, what are really nice, well-isolated units, and what are just multi-unit activity. You would typically do this in Phi, which is a software to look at all of your data and your units, unit by unit. And one thing that's really important here, because you're doing this just yourself, or several people might be doing this on the same data sets that you then want to publish, you really want to have a particular workflow that you're consistent in sticking to, because it can be very easy for you to get tired and slightly change your criteria and have some bugs and artifacts that are just due to you being tired or different people looking at the data in different ways. So here's an example of a workflow that you could follow. Typically, you would look at each unit and first try and determine, does this look like noise? Looking at the waveforms as we talked about previously. And then you would try and understand if there are false positive or false negative spikes. So here's the software Phi. I'm going to try to open it up. Sure. Okay, hopefully you'll see that. Oops. So this software loads all of your data, and there are different views that we've already seen previously, but that I'm going to briefly explain here. So you select each of your units in this cluster view, and here we have a selected unit with its waveform plotted here. So this is a subsample, a few raw waveforms that have been extracted. You can also show the mean of those waveforms or what the template looks like. You also see this unit's autocurlogram in this view. And these lines, again, correspond to the unit's mean firing rate and plus minus 1 millisecond window. This view shows you all your raw data. And you can see one of the spikes for this unit here in blue in the raw data. And you can toggle and look at different spikes for that unit to try and get a sense of what the raw data looks like. This view plots a proxy for each spike's amplitude in the y-axis and time in the x-axis. And you can see the histogram of the distribution of amplitudes here, which allows you to visually kind of estimate what percentage of your spikes are missing. And so, for example, for this unit, if we go back to looking at the raw waveforms, the waveforms do look neuronal. So we do see spatial decay, and there is one nice trough and peak. It does have a neuronal shape. So this is probably not noise. This unit also has a clean refractory period. And there's consistent spiking throughout the recording. It doesn't seem like a large portion of spikes are missing due to drift, or that a lot of the spikes have been cut off and are not detected. So this is probably a good neuron. And then we would try to look at more similar neurons by pressing spacebar. This software brings it up, and try to assess whether some of the spikes have been wrongly classified into another unit. And in this case, if we look at the next most similar unit, they seem to be rather different. So it's probably not true. Okay, if I look at just the next few units. So this is an example of a unit, as you can see, where a lot of the spikes have not been detected. It's probably a unit that's further away from the probe, and this we would probably classify as multi-unit activity. And finally, I'll see if I can find an example of noise. Maybe not. Well, maybe I'll just leave it at that. Okay, so then I think Noam is going to talk about how to do this in an automated way, which quality metrics you can use that corresponds to which step of the manual curation process.
All right, hi everyone. Can everyone see my screen? Good. Okay, great. All right, so thanks, Julie. So I'll be talking about why use automated quality metrics. So I just want to, before I start, kind of say that these two approaches really go hand in hand. I don't recommend only using automatic quality metrics without ever looking at your data, but rather using some manual curation and then to inform which quality metrics will be most useful for answering your scientific questions. And I'll kind of keep reminding you of that throughout this talk. All right, but why do we want to use automated quality metrics? So at this point, due to the huge increase in the number of neurons that you're able to record from with NeuroPixels and with other recording technologies, it takes a long time to look through, as Julie was just showing in that Phi demo, to look through many, many recordings by hand. So we basically kind of aid this with some metrics that we can use to assess the quality of our neural data, of our sorted neural data. All right, and so before I get started, I just want to sort of make a general point that there's not one agreed upon good neuron. There's some continuum between complete junk, just noise that's not even a neuron, and the best possible neurons. And different people may care about different properties of neurons and may define a good neuron in different ways. So given all of that, don't forget your raw data. Make sure you're not only blindly using automated quality metrics, but now we'll start kind of talking about what those automated quality metrics are. Okay, so we can use quality metrics to basically assess spike sorting goodness and then automatically curate your data. So they allow us to quantitatively assess the goodness of a spike sorting output. And then the definition of a good unit depends a little bit on your scientific question. So for an example here, if you care, if you're doing maybe a lot of population analyses and you don't care about exact spike timing of one specific neuron, maybe you're all right including some multi-unit activity in your recording. But if you do care specifically about really well isolated neurons and you want to make sure that you're not getting any false negatives, any missing spikes, then you will want to use some quality metrics that address that specifically. All right, and so yeah, so we'll go through some different types of quality metrics. And these are basically based on our general neuroscientific knowledge of what good units should look like. Okay, and then one more sort of intro slide, some general pros and cons here. First, doing automated quality metrics can remove some human bias from doing manual curation. So as Julie mentioned, even when you're just one person doing manual curation, you can get tired. Your sort of mental thresholds of what a good unit is can shift across time. If there are multiple people doing manual curation, they may be doing it somewhat differently. So you can see some biases and differences here. And of course, it saves you a lot of time. Some cons to be aware of. So generally, quality metrics are defined at the unit level, not at the spike level. So basically, we say a unit is good or a unit is bad, passes or fails. So if you have kind of more specific questions about individual spikes, then you may have to kind of dig in and do more manual curation. And then lastly, and I'll mention this throughout when I describe some of the quality metrics, the thresholds or the exact quality metric criteria that you're setting may vary depending on the brain region you're recording from, the species you're recording in, brain state, the context of the experiment, and other factors. So you do want to think carefully about which quality metrics you're using and make sure that these are not confounds in what you're doing. All right, so I'll just sort of start with a bit of an overview of general characteristics of what a good neuron looks like. And these are very in line with what Julie presented. But again, which characteristics specifically you care about might depend on your scientific question, and there's not really one established answer for what a good neuron is. All right, so a few of these characteristics: a clean refractory period, so few refractory period violations, large amplitude, dissimilar waveform to anything else nearby, meaning you don't have two neurons that are counted as one, a consistent waveform across time, and then spikes not missing, not lost below the detection threshold. Okay. So I'll start out, and I'm basically just going to kind of go through a list of some available quality metrics. This is an incomplete list. There are certainly other people who are using other quality metrics. And at the end, I have these resources listed here, but at the end, I have sort of a slide with all of the links, and I'll make that available to you guys. So, so you can look at all the documentation for all of these already computed metrics and see what makes sense for you. All right, so to address the sort of first part of Julie's manual curation steps of getting rid of complete noise, the main thing that you can use in terms of quality metrics is waveform classification. So you can find, there's some algorithms for finding non-semantic or noise waveforms. You can also do some quality metrics based on, for example, the number of peaks and troughs, and there are a few other types of quality metrics available there. One other thing that generally is thought of to be for false positives and not really for noise, but you can sometimes use it to inform whether you have noisy neurons, is ISI or refractory period violations. So if you have a completely flat, and this is not completely flat, but a kind of a somewhat of an example, refractory period that has a lot of spikes within that refractory period, you might think that this is actually not a neuron at all. It might be noise. So mostly waveform classification for this, but you can also sometimes see this with ISI violation metrics. All right, so some more typically used quality metrics. So one, people will sometimes use a firing rate threshold. So this is just the mean firing rate, either in one part of your recording or across the entire recording. Some potential issues here: we know that firing rate is very variable across the brain and across different cell types. So setting one specific firing rate threshold can sometimes mean that you miss neurons that might be very well isolated, just have low firing rates, like ground truth low firing rates. And here's just a plot from the Allen Institute of distributions of firing rates in 4 different brain areas. And you can see that there are differences in the firing rates across these different brain areas. All right, you can also use an amplitude threshold, but a similar kind of potential issue for that is that some small amplitude units could be indeed well isolated and you're just missing them if you have an amplitude threshold. Typically, people will just do a peak to trough, peak minus trough to find the amplitude of the neuron, and then use a threshold for either the mean or the median of the amplitude. All right, next, and I spoke about this a little bit before, is ISI violations, so refractory period violations. And so typically there's a few different types of this metric. Typically people will just compute the percent contamination, basically a measure of how many refractory period violations you have relative to the firing rate of the neuron. There's a couple of issues here. So you are assuming a set refractory period here. We actually developed a metric that gets rid of that assumption, and I'll link the code to it at the end of the talk that we think works pretty well for getting rid of that assumption that refractory periods are the same across the brain. Also, this doesn't work quite as well for low firing rate neurons. So depending on whether or not you have a firing rate threshold, you may have to keep that in mind. All right, and then isolation distance. So this is basically a metric of how well isolated your neurons are. So there are several different types of isolation distance metrics. A lot of people use this Mahalanobis distance metric. You compute basically the distance to the nearest cluster. You do have to be careful because if your neurons have drift, you might see the variance change across the recording in a way such that your isolation distance will be computed incorrectly or will be not fully informative. So you do have to be careful about that when you're just sort of if you're blindly implementing that. There's also many other isolation distance metrics that I just don't have time to go into. So you can compute the D prime, you can do an L ratio. There's a few others. These are all listed in kind of the resources I'll list later on. Okay, and then a few final ones. So presence ratio is a metric that tells you about whether spikes are present throughout your entire recording. So if you just look at a plot of all of the spikes across the time of your recording, you can compute how much of the time of the recording you do see spikes. So for example, you have a neuron that spikes the first half of the recording, but then stops spiking, then you may not want to include that neuron. The caveat here is that this might be a problem for neurons that are actually encoding a time-dependent variable. So for example, if you're interested in thinking about motivation or satiety, reward signaling, you may see that an animal that's working for an entire experiment, their motivation or their satiety increases or decreases depending, and a neuron that's encoding that variable may have a low presence ratio because it may only fire, for example, when the mouse is motivated to do the task. All right, and then amplitude cutoff, which Julie mentioned a bit. So this is basically a missed spikes estimate based on the amplitude histogram of the neuron. So that's, I'm just showing a screenshot from Phi here. And you can see that this distribution of the neuron's amplitudes here is kind of sharply cut off at the bottom here. So there are spikes that were missed that fell below the detection threshold here. And so you may not want to include this because you're not getting a full picture of all of the spikes of your neuron. One issue with this is that this specific missed spikes estimate that's typically implemented does have a Gaussian assumption on the actual distribution here. So there's one alternative that I'll share in the resources page at the end, but there's just sort of pros and cons to that. And then lastly, you can have some drift metrics, so you can compute the kind of maximum change in spike depth throughout the recording and the cumulative drift throughout the recording. But I do want to just have a caveat there that sometimes when you have merges and splits, that look like drift. So for example, here, Phi classified this as 2 different neurons, but it may just be one single neuron that was drifting, and you will need to, that's sort of tricky to do with automated curation. So you may need to go in, define, and do a manual merge or split, depending on the case for that. Okay. Before I end, I'm sorry, we're running out of time, but I just want to remind you to think carefully about the scientific questions that you're trying to address and use that to inform which quality metrics are most important for you. And as always, do look through your data. Don't just sort of blindly apply some quality metrics and assume that they're giving you the answers that you want. So here I'm showing an example plot. The specific scientific question here doesn't matter, but they're basically plotting some variable of interest. Here is spatial information in the population of neurons they recorded. And what they wanted to check is that the effect that they see, and here it doesn't matter the details, but the effect is basically that there's this positive amount of spatial information in their population. They wanted to check that that isn't only true, isn't a confound, and isn't only true because of poorly isolated neurons. And so what they did is they plotted all of their neurons, not just the ones that pass, as a function of the isolation distance, which is one of these quality metrics. And what you see is that even for the well-isolated neurons, these ones towards the right of the x-axis, they still see the effect that they were showing. So it's just sort of these types of sanity checks to make sure that the neurons that you're including in your analysis are high quality neurons. You can also do some specific waveform examination. So one common thing people do is just plot all of the template waveforms for each neuron. And then you can plot all of the noise ones and just sort of check that there's not waveforms getting through that you don't want to get through. All right. Okay, and so then lastly, this is just some resources, and of course we'll share this. So the Allen Institute has some really wonderful documentation and code available for all of the quality metrics that they use. They also include which thresholds they have as default and which quality metrics in general they use as default, but they compute many more quality metrics. And the list that I gave today is very similar to the list from the Allen Institute, so do check that out. Spike Interface has a very similar module on that. Both of these are in Python code, and they're all really well documented and really useful. Julie from Cortex Lab also has this great quality metrics guide on this GitHub repo. So this is code in MATLAB. It's really fantastic. The IBL and Steinmetz lab have a sliding refractory period, which I briefly mentioned, which doesn't use this 2 millisecond assumption. I've linked the code for that. That's both in MATLAB and Python. And then the IBL uses a few metrics also for just all of their data. And we have a white paper describing all of that, including all of our spike sorting pipeline as well. So feel free to take a look at that. All right, and that's all I have. So we can just open it up to a discussion and questions at this point with Julie and Alessio and myself. Thanks.